Here's my depiction of photosynthesis and cellular respiration. Photosynthesis starts off in the chloroplast in the phospholipid bilayer of the thylakoids. And in that phospholipid bilayer, sunlight hits a photostem number 2, and that excites electrons, which hit the main chloroplast and which hits the main chlorophyll, and then they bump up and to the primary electron receptor, and they are channeled through the electron transport chain. In that excitement, 2 molecules of water are broken apart into oxygen and hydrogen, and hydrogen is built up in the inner thylakoid to build the pH gradient, and the oxygen is used to pull the electrons through the chain, since it's electronegative. And the electrons first spend their energy in the cytochrome complex, which pumps additional hydrogen in from the stroma. And then the de-energized electrons then go to the photostem 1, where 700 wavelength light is accepted, and it re-excites the electrons, and so it allows them to reduce NADP through NADP reductase, making NADPH. That's later used in the light-independent reactions in the stroma. And then the hydrogen gradient that's built up here is used by ATP synthase. The hydrogen is pumped out to make ATP in a process called chemiosmosis. And that ATP and NADPH go down to the Calvin cycle, where there are 3 stages: fixation, where rubisco attaches carbon to RUBP, making it a molecule that breaks down really fast, and then biphosphate, which is 3 carbons. Then enters reduction, where 6 ATP and 6 NADPH are used to make biphosphoglycerate, which is also 3 carbon. Then it makes 6 molecules of G3P. 1 is given off to make glucose, and such. And then 5 continue on and are acted on by another 3 ATP, turning them back into rubisco, or RUBP, which is 5 carbon. And so it can continue the cycle. Then the glucose, or the half of a glucose, is given its other half, 2G3P. That glucose is sent on to cellular respiration. Starting with glycolysis, the glucose is acted on by ATP. This is in the cytoplasm. Glucose is acted on by ATP, making glucose 6 carbon. Another ATP is used on it to make fructose 6 carbon biphosphate, and that breaks into 2 G3P, which make 2 ATP and 1 NADPH each, or 1 NADH each. Then it makes pyruvate, and starts the pyruvate oxidation, where 1 NADH is made each, 1 CO2 is released each, and a coenzyme is attached each to make acetylcoenzyme A. 2 of them, since there's 2 G3P. And then after the glycolysis and pyruvate oxidation, it enters the Krebs cycle, where acetylcoenzyme A acts like rubisco in the light-independent reactions of photosynthesis. It gives energy back to oxaloacetate, turning it back into citrate, which makes an NADH, which then this citrate, which is 6 carbon, makes NADH, becomes ketoglutarate, 5 carbon. NADH is made, ATP is made, then it becomes succinine, and another NADH is made, FADH2 is made, then it becomes oxaloacetate, which is 4 carbon, succinate's also 4 carbon, and the acetyl coenzyme builds back up its potential energy back into citrate, keeping that cycle going. And those electron acceptors that are made in the Krebs cycle go on to oxidative phosphorylation, where the first protein takes NADH, reverts it to NAD+, second one takes FADH2 back and transfers it back to FAD+. It takes its hydrogens, pumps it into the inner membrane space, and all this is happening in the mitochondria. It pumps it into the inner membrane space, and the electrons go through the chain to be eventually pulled through and accepted by oxygen to make water. And those hydrogens that are up here are pumped through ATP synthase to make, in the process of chemiosmosis, to make ATP. 3 are made from every NADH, 2 ATP are made from every FADH, and you should end up with 38 molecules of ATP from cellular respiration. And that's how you get from light to usable energy.